so that patients can be treated. If you are successful in your mission, you will be able to prevent a full-scale epidemic among the Kuppelfangs here on the exoplanet Astakos 4. Click Continue to find out why you are here on Astakos 4 and what Kuppelfangs are.
This simulation uses voiceovers.
Using the lab pad is an important way to get the most out of this simulation. Perfect. Then let's get started. You are on Astakos 4, an exoplanet in the Milky Way where we have found complex and fascinating life forms. Let's step out of the lab and take a moment to explore this beautiful planet. Human pioneers have formed small settlements on Astakos 4. The settlers are mainly scientists who study the planet's physical environment and living creatures. One of the creatures here on Astakos 4 is called the Kuppelfang. They have intricate shells on their backs and digest rocks. You can see a group of baby Kuppelfangs here. The Astakos 4 ecologist Francisco is studying these fascinating beings. Unfortunately, he found an unwell baby Kuppelfang 2 days ago. Since then, more Kuppelfangs have fallen ill with similar symptoms. The Kuppelfangs look pale, have a fever, are lethargic, and some of them throw up or have diarrhea. If you look carefully at the environment, you will see lots of Kuppelfang vomit. Francisco knows that having sick Kuppelfangs will affect both the species and the ecosystem on Astakos 4. He urgently needs your help to find out what is wrong with the Kuppelfangs to stop the spread of the disease.
Based on what you know about the Kuppelfang's illness, fever, fatigue, vomiting, and diarrhea, what is the most likely cause? Click the View Theory button if you need guidance with the question.
Option A, infectious disease. Option B, genetic disease. Option C, injury. Option D, environmental factors. Good guess. Fever and fatigue are signs of infection. Vomiting and diarrhea can also be a sign of infection in the gastrointestinal tract. The disease is also spreading quickly through the population. This suggests that the disease can pass from one individual to the next, but to be absolutely sure, you will need to do some tests in the lab. Take a sample from one of the sick Kuppelfangs to take back to the lab. Great job! We are not sure whether all the Kuppelfangs have the same illness. I will take another sample and analyze it in the lab alongside you. Let's take the samples back to the lab now to run some tests. We will be leaving behind a camera so you can check on the Kuppelfangs from the comfort of the lab. Pick up a lab coat from the rack to your right.
Well done. Now, find the glove box on the cab.
Go to the wall screen to check on the Kuppelfangs via the camera we left behind. More and more Kuppelfangs seem to be getting sick. Microbiologists use lots of different techniques to identify unknown bacteria. I have sent a sample of the bacteria to Earth for a complete analysis, but we might not have time to wait for the results. We do not have all of the possible techniques for identifying unknown bacteria available here on Astacos 4, but I think together we can find out what is making the Kuppelfangs ill. Click Continue to help me get started on our analysis so we can treat the Kuppelfangs as soon as possible. Explore the workbenches in the lab to find out more about the techniques you can use to identify bacteria. Interact with the lab pad if you want to read the text. Differential media use the metabolic and biochemical characteristics of bacteria to distinguish between bacterial species based on how they look. Changes to the colony morphology or medium indicate whether a bacterium is able to ferment sugars or has certain enzymes. Hover over some of the tools and reagents on this workbench to find out what they are used for. Many bacterial metabolic processes are driven by enzymes and vary widely across bacterial species. Biochemical tests use this variation to identify bacteria. You can find out what the tools and reagents on this cabinet are used for by hovering over them. Staining is a technique used to enhance contrast in biological samples before looking at them through a microscope. Once bacteria are stained, we can study their size, shape, and arrangement. Through staining, we can also visualize cell structures. Hover over some of the tools and reagents on this workbench to find out what they are used for. Great! Now that you know your way around the lab, let's look at the experiments that you will do here today. Remember that you can always click on Theory to check out the Theory tab, or hover over an object in the lab if you forget what it is used for.
Go to the interactive poster next to the microbiology workbench. Fill in the blanks to work out which results you can expect from each test. You can click the View Image button to display the incomplete interactive poster in the lab pad.
Let me highlight the relevant objects for you. That's not quite right. Check out the differential staining theory page if you want to know more. Not quite. Check out the differential media theory page if you want to know more. Almost there. Check out the biochemical identification theory page if you want to know more. Click on the lab pad for more information.
Excellent. Throughout the simulation, you can come back to this poster to interpret your results and work towards a diagnosis. You can also find this overview in the Media tab or clicking the View Image button.
Go to the microscopy workbench to start experimenting.
The Gram stain is the most common stain used in clinical microbiology. Cells with a thick peptidoglycan cell wall stain purple, known as gram-positive, shown with a plus symbol, while cells without a thick peptidoglycan cell wall stain pink, known as gram-negative, shown with a minus symbol. Because it makes a distinction between two classes of bacteria, the Gram stain is called a differential stain. Since peptidoglycan is a target for some antimicrobial drugs, the Gram stain also provides some information on antimicrobial drug susceptibility.
Depending on the result of the Gram stain, information about the symptoms and environment of the patient, a microbiologist might do additional stains, such as the endospore stain or the acid-fast stain. Would you like to learn more about differential staining? Option A.
Bacteria come in many shapes and sizes, but always maintain their shape thanks to their rigid cell wall. The outer layer of bacterial cells, the bacterial envelope, consists of a plasma membrane and a cell wall, which protect the cell from the extracellular environment and give it shape. Some bacteria contain extracellular features, such as a capsule, fimbriae, pili, and flagella for adherence, movement, attachment, and transferring genetic content. Inside the cell, the bacterial DNA is located in the nucleoid. Like eukaryotes, bacteria have ribosomes that transcribe the DNA, but they are 70S rather than 80S. Bacteria can also contain circular DNA molecules called plasmids, which can be transferred between bacteria and often give a selective advantage. Click the View Image button to display the full-screen image in the lab pad.
Would you like to learn more about
The Gram stain differentiates bacteria based on their cell walls. Crystal violet stains peptidoglycan in the cell wall purple. In gram-positive organisms, the cell wall has a thick layer of peptidoglycan, which retains the stain. In gram-negative bacteria, the peptidoglycan is thinner and does not retain the purple stain. These cells are visualized by the pink counterstain safranin. Gram-positive cells also take up safranin, but this is not visible due to the dark purple crystal violet. The acid-fast stain stains another component of the membrane called mycolic acids. These are present in mycobacteria, including Mycobacterium tuberculosis, which causes tuberculosis. Acid-fast bacteria retain the red color, while non-acid-fast bacteria are counterstained blue. Endospores, tough dormant structures produced by some bacteria to reactivate under favorable circumstances, are difficult to stain. Malachite green stains the cortex of the endospore green, but is not retained by the bacterial cells. These are counterstained by safranin, just like the gram stain. Click the View Image button to display the full screen image in the lab pad.
Would you like to learn more? You can check out the Theory tab on your...
Which stains are used to visualize structures in the bacterial cell wall? You can click the View Theory button to get some guidance with the question.
Gram-tastic. The gram stain stains peptidog lichen, and the acid-fast stain stains mycolic acid, which are both in the cell wall.
What color do endospores and bacteria stain in an endospore stain? Click the View Image button to...
That's right. You are ready to start your own experiments. I have already performed the bacterial staining procedure for you. You just need to interpret the results of the gram stain. To remind yourself of the staining procedure, you can always check out the gram staining simulation. If you need more information, pick up the gram-stained sample and insert it into the microscope. Turn to your left and click the screen to see the image of the sample.
What is the morphology of your bacteria? Click the View Image button to display full screen the image in the lab pad.
Option A, gram-positive coccus. Option B, gram-negative bacillus. Option C, gram-negative coccus. Option D, gram-positive bacillus. Great job! You've got the size and shape of it. Go to the wall screen to see which bacteria might be making the couple fangs ill.
Here, you can see the characteristics of some bacteria that might have infected the Kupelfangs. They are carried by the humans on Astacos 4 and could cause symptoms that match those of the Kupelfangs. Based on the staining results, you have already eliminated 2 candidates. The names of those bacteria have gone gray. Great work. If you are looking at the lab pad right now, remember that you can slide it down at any time by clicking on the background. You can always come back here to study your suspects. Click the View Image button to...
The gram stain provides a lot of clinically useful information, but it won't give you all the information you need for identification and treatment. About which of these do you not learn anything from the gram stain? Option A, structure of the cell wall. Option B, bacterial morphology.
That's right. Gram staining provides information about the structure of bacterial cell walls and bacterial morphology. Antimicrobial drugs work by disrupting bacterial cell walls, meaning that gram staining can also show whether bacteria are susceptible to these drugs.
As you have seen, microscopy can show the cell shape and cell structures. Not all characteristics that we use for identification of unknown bacteria can be seen under the microscope. Techniques such as immunoassays, mass spectrometry, and nucleic acid-based techniques are increasingly used in clinical microbiology labs. Immunoassays use antibodies, proteins that bind to very specific targets known as antigens. Antibodies specific for pathogenic bacteria or their products can be used to identify these bacteria. You can learn more about immunoassays from the ELISA simulation. Mass spectrometry, MS, is an analytical tool that measures the mass of molecules. This can be used to determine the molecular formula. Thus, MS can be used to determine the amino acid sequence of peptides in a microbiology sample. These results can be used to search a database or compared against the translated genomes of bacteria. Immunoassays, mass spectrometry, and nucleic acid-based methods for identifying bacteria can be very specific and do not rely on growing bacteria in the lab like the methods you are using today. Differential media and biochemical tests use the metabolic and biochemical characteristics of bacteria to identify the species. Let's find out how you can use these techniques to help save the Kupelfangs at the microbiology workbench. Go to the microbiology workbench. Culturing is the practice of growing bacteria in a lab. In order to grow, the bacteria must be provided with the correct temperature, nutrients, and other factors. Nutrients are provided by the culture medium. There are 4 main types of culture medium. Nutritive media support the growth of most bacteria without giving any particular species an advantage. Enrichment media provide specific nutrients that selectively support the growth of particular bacteria over others. Differential media contain factors that allow colonies of one type of bacteria to be distinguished from another, growing on the same agar plate based on their appearance. Selective media contain inhibitory agents, such as antimicrobial drugs, bile salts, and acids that allow only the selected bacteria to grow. Differential and selective media can both be used to obtain pure cultures from a single colony. The same medium can be both selective and differential. For example, McConkey agar is selective for gram-negative bacteria and differential for lactose fermentation.
Which type of medium supports the growth of the widest range of bacteria? Click the View Theory button if you need some guidance with the question.
Option A, differential. Option B, nutritive. Option C, enrichment. Option D, selective. Excellent. A nutritive medium is a general-purpose medium with different nutrients that also lacks any agents that could inhibit growth. It therefore supports the growth of many different bacteria. Bacteria form colonies on solid media. Microbiologists use the streak plating technique to ensure that there are individual colonies on the plate. Colony morphology and motility can help to identify bacteria. Individual colonies also allow microbiologists to obtain a pure culture. This is a culture generated from a single colony in which all the bacteria are identical. The pure culture can be used to do further tests. Why is it important to have individual colonies? Option A, to look at colony morphology. Option B, to look at motility. Option C, to obtain a pure culture. Option D, all of these statements are correct. That's right, you are becoming a microbiology genius. McConkey agar is selective for gram-negative bacteria and differential for lactose-fermenting bacteria. Blood agar is a nutritive medium, differential for hemolysis. Based on what you know about the candidates on the wall screen, which type of differential medium will give you the most information? Option A. Let's test it. McConkey agar should tell you whether your bacteria are P. mirabilis, lactose negative, or either E. coli or K. aerogenes, both lactose positive. You have already excluded both B. hemolytic organisms, but you can also check for the swarming motility typical for P. mirabilis with blood agar. I think we should try both media. Let's start with McConkey agar. Pick up a McConkey agar plate from the stack and place it next to the Bunsen burner. Turn on the Bunsen burner and sterilize the wire loop. Your sample is on the workbench. Use good aseptic technique to set up a culture of your sample. Let's put your plate in the incubator on your right to grow the bacteria. Nothing to pick up or drop. Close the incubator to start the incubation. While you were streaking out the sample on McConkey agar, I have streaked it on blood agar and placed that plate in the incubator too. These need to incubate for 24 hours. Luckily, we are in a virtual lab and can speed up time. While we wait, let's see how well you understand differential media.
What is the difference between selective and differential media? Click the View Theory button if you need guidance with the question.
Option A. Differential media inhibit the growth of certain bacteria. Selective media distinguish different bacteria from each other. Option B. Selective media. Excellent! You are the media master. What would you expect to see if you cultured gram-positive lactose-fermenting bacteria on McConkey agar? Option A. Yellow agar with pale colonies. Option B. Great job! You are starting to differentiate your differential media. Bile salts and crystal violet in McConkey agar inhibit the growth of gram-positive bacteria. Take the McConkey agar plate out of the incubator and put it on the workbench, then close the incubator.
Inspect the plate. Are your bacteria able to ferment lactose? You can lower the lab pad or click the View Image button to inspect the plate.
Option A.
Yes,
Great job! The fermentation of lactose lowers the pH of the medium, which you can see as pink colonies thanks to the pH.
Now let's have a look at the blood agar plate I cultured for you. Click the View Image button to display the full-screen version of the image in the lab pad. Identify the activity of your sample on blood agar.
Option A.
Alpha hemolytic.
Option B.
Beta hemolytic.
Bloody amazing! Your bacteria do not lyse the red blood cells in the blood agar. Throw your sample in the waste bin to your left to dispose of it safely. Let's look at your progress on the wall screen.
You are making progress. There are still two possibilities. If you can find out the biochemical properties of your sample, you will find out what is making the Kupelfangs ill.
Go back to the microbiology workbench to do some biochemical tests. Biochemical tests use variation in metabolic processes and enzymatic activity to identify bacteria. A single biochemical test is not sufficient for identification, but different biochemical tests can be critically combined to successfully identify bacteria. Biochemical tests can also be critically combined with other methods, as you are doing now. Would you like to learn more about biochemical tests? Like all cells, bacteria take up nutrients from the environment and transform them into energy and cellular components. Enzymes, proteins with catalytic activity, are required to carry out many of the reactions necessary for metabolism. Metabolic processes and enzymatic characteristics vary between bacterial strains and species. Such differences are exploited by differential or selective media and biochemical tests. Would you like to learn more about biochemical tests? Option:
Closely related organisms can be differentiated using a series of biochemical tests that identify various metabolic properties of different bacterial species. A single biochemical test is usually not sufficient to identify an unknown bacteria, but biochemical tests can be critically combined with each other or with other methods of bacterial identification. The catalase test is designed to detect the presence of the enzyme catalase, which converts hydrogen peroxide into oxygen and water. Because hydrogen peroxide can damage the cell, catalase protects the cell. It is present in most aerobic bacteria. The presence of catalase can be determined by the production of bubbles of gas when bacterial culture is added to hydrogen peroxide. No bubbles are produced in catalase-negative organisms. The oxidase test detects the presence of cytochrome C oxidase through a color change. Cytochrome C oxidase is an enzyme present in many aerobic bacteria. It catalyzes the transport of electrons from electron donors' donor compounds to electron acceptors in the energy-producing electron transport chain. In the oxidase test, the enzyme oxidizes a colorless or light pink reagent to a blue color. The enzyme urease catalyzes the reaction of urea into ammonia, water, and carbon dioxide. In the urease test, presence of the enzyme can be determined by adding urea to the medium and detecting ammonia through the associated increase in pH. The indole test determines the ability of bacteria to produce indole from tryptophan by various enzymes. Indole can be detected in a bacterial culturing by adding it to p-dimethylaminocinnamaldehyde, DMACA, which results in a blue color. If no indole is present, DMACA remains colorless or becomes light pink. Click the View Image button to display the full screen image in the lab pad.
Would you like to learn more about You can check out the Theory tab on your lab pad at any time to learn more about biochemical tests.
Which molecule do indole-positive bacteria cleave to form indole?
That's right. Various enzymes convert tryptophan into indole in indole-positive organisms. Which enzyme is part of the electron transport chain in many aerobic bacteria? Option A. Great job. Cytochrome C oxidase is part of the electron transport chain in some aerobes. Remember that some aerobic organisms use a different enzyme, so there is not a complete correlation between oxidase and aerobic respiration. Based on your results so far and what you know about the candidates, which biochemical test do you think you should use? Option A, indole test. Option B, catalase test. Option C, oxidase test. Actually, I think we should do all three. I tricked you a bit there. Some of the tests will be necessary to distinguish between the microbes, while others will help verify our earlier results. After all, we need to get this right to help the couple fangs. We will start with the catalase test and then continue with the other biochemical tests. Use the blue dropper to add hydrogen peroxide to the test tube.
Click the blue dropper instead to pick it up.
Pick up some sample from the sample tube and transfer it to the test tube with hydrogen peroxide. Pick up the test tube to check whether your bacteria express the enzyme catalase. Look carefully as the bubbles may be hard to see. Do your bacteria have the enzyme catalase? Option A.
Yes, catalase positive.
Option B.
No, catalase negative.
Great job! Let's clean up so we can continue with some more biochemical tests. I have already tidied away the hydrogen peroxide and fetched the reagents you need for your next experiment for you. Sterilize the wire loop, put it back, and dispose of the contaminated test tube in the waste bin. I have already put away the hydrogen peroxide for you. All right, let's do the oxidase test now. Make sure the Bunsen burner is on and place the test strip in the sterile work area close to the Bunsen burner and sterilize the wire loop. Please put down the item you are holding first. Use aseptic technique to pick up some sample from the sample tube and add it to the test strip. Sterilize the wire loop and pick up the test strip to inspect it.
Do your bacteria have the enzyme cytochrome oxidase? Click the View Image button to display an image of the possible oxidase test results in the lab pad.
Option A.
Yes, oxidase positive.
Option B.
No, oxidase negative.
You are so fast, you must be a catalyst. Put the test strip in the waste bin and turn off the Bunsen burner.
I also performed the indole test on your sample. Click the View Image button to display the image full screen in the lab pad. Are the bacteria in your sample able to cleave indole from tryptophan?
Option A.
Yes, indole positive.
Option B.
No, indole negative.
All right, let's see what that means. Go to the wall screen and use the buttons to inform Francisco of the name of the bacteria that you have identified.
Great work! Your results have been sent to Francisco, who can now treat the Kupelfangs. It will take the Kupelfangs a few days to get better. Let's see whether they improve. You
are now a microbiology expert. I have done all the tests on the other sample alongside you. Will you help me to interpret the results? All right, let's start with my staining results. What type of bacteria are these? Option A. You have a trained eye.
I also plated this sample on McConkey and blood agar. Identify the lactose fermentation and hemolytic activity of these bacteria. Click the View Image button to display the image full screen in the lab pad.
Option A.
Lactose positive, gamma hemolytic.
Option B.
Lactose positive, beta hemolytic.
Option C.
Lactose negative, gamma hemolytic.
Option D. Double check your results. It's important.
I also plated this sample on McConkey and blood agar. Identify the lactose ferment.
Double check your results.
I also plated.
You are the media master.
How about these biochemical test results? Click the View Image button to display the image full screen in the lab pad.
Option A.
Catalase positive.
Nearly there, so it is important you...
How about these biochemical...
Wow, you really are a microbiology expert. Let's see whether the treatment had an effect. Wow, those Kupelfangs have made a full recovery. Thanks to your growing microbiology skills, you identified the bacteria very quickly. This allowed Francisco to use the correct treatment for the infection. It looks like we were just in time with our treatment.
The methods you use today rely on being able to grow the bacteria in the lab in a pure culture and high enough number. Some bacteria have ecological niches that are difficult to replicate in the lab and cannot be cultured. Using nucleic acid-based techniques allow microbiologists to study these bacteria. It can also be used to identify bacteria. In a hybridization assay, DNA or RNA from the bacteria is mixed with known DNA or RNA sequences called probes. When the bacterial target sequences bind to the known probes, a fluorescent signal appears. Since you know to which bacteria each probe corresponds, you can identify the bacteria in your sample. Click the View Image button to display the image full screen in the lab pad.
It sounds like your colleague, Dr. Y. Pestis, is calling from Earth with their results. Go to
Greetings, esteemed colleague. I have received your sample and extracted DNA from the bacteria to perform a nucleic acid-based hybridization assay. I was able to see that the Kupelfang you took a sample from was infected with K. aerogenes. Dr. 1 took a sample from another Kupelfang that turned out to be infected with another bacterium. Thanks to both of you, all the Kupelfangs have now been treated by Francisco. You did a stellar job there on Astacos 4.
The expert agrees with you. Great news. These bacteria, along with many others, live in the human gut where they do not cause any harm and even help digest food. They are part of the human microbiome, bacteria that live in and on the human body. Such bacteria are sometimes known as commensals. Since the bacteria are foreign, even alien, to the Kupelfangs, they had no defenses against the invasion and colonization of their gastrointestinal tract. Bacteria that are commensal to humans can be pathogenic for other creatures. In hospitals, you could come across similar scenarios. For example, immunocompromised patients are vulnerable to infection with bacteria that would be harmless to healthy people, or a bacterium that does not cause disease in livestock could make humans ill. Think, for example, of Salmonella infection. Thanks to you, the bacteria that had infected the Kupelfangs have been identified. Now that the Kupelfangs are back to their usual rocking selves, the humans here on Astacos 4 must be much more careful to avoid giving these bacteria to the Kupelfangs. I feel a little unwell myself, so I will go upgrade my antivirus software. Goodbye. Let me ask you one last question.